Okay, good news. I've managed to fix it. Can you hear me, guys? Okay, the good news is I've managed to fix it. Now let's go for it. All right, what we're going to do today is last part of excretion, which some people have trouble with, and then if we do that last part of excretion, then we master reproduction in both humans and in plants. Okay, we just do the whole thing. So I'm going to do the last part of excretion, and then I'm going to do with reproduction one time, and let's see how it goes. So when we say a person understands excretion, right? This chapter of excretion, what is it that you'd need to know for you to say, for us to say you understand excretion? What you need to know is you need to know the kidney basically and the structure. Excretory system is good. They can ask you, but most of the questions will be on the kidney and the nephron. So you should know the nephron and the jobs it does and how it does ultrafiltration and selective reabsorption. You must know that. That's good. When you know that somebody needs to mute the mic, somebody needs to mute the mic. Who is that? Let me help. Make sure your mics are muted, guys. Okay. I think it's better now. So what I was saying was, um, you need to make sure that you have me right, and then I'm going to just write, guys. You need to just make sure you know selective reabsorption. This thing called the nephron, which is a very small thing. There are millions in each kidney, about 1 million of them in each kidney. So you must know. Okay, you must know the structure of the kidney, and then you must know the nephron, and then you must know, of course, when I say the excretory system is the two kidneys, the ureter, the bladder, the urethra. Okay, and it's also good to know the valves, the blood vessels. It's also good to know what is renal vein, what is renal artery, stuff like that. What we do know is that nothing called a vein will ever bring you blood. It will always go with the blood back to the heart. Veins are loyal to the heart, if we can say that. Arteries take blood from the heart. Veins will always take blood back to the heart. Are we together? So when we're saying if the renal vein is taking blood back to the heart, and then the two kidneys, you should know your two kidneys. Each one has a million nephrons, and you have the ureter, then urethra after. So ureter bladder, then urethra bladder keeps the urine, and then it now takes it outside through the urethra. Then here is the kidney, what it looks like. If you look at the side view, we know that you have two. Yes, we know. We know that you have two. What is it? Inside it is a nephron. You should know the different parts of the nephron. You should know the glomerulus, the proximal, the distal, the loop of Henle, the collecting duct. That's good. You should know these parts. So that's actually good. So, and also you should know the Bowman's capsule, of course. So glomerulus, this is all about veins. They are bringing and the arteries. They are bringing. They're very very small blood vessels. We call them arterioles. They are coming together and tying themselves into knot. And when we say they are going into knots, it means they are trying to keep round to stay be thinner and thinner and thinner and thinner. They want to be very long inside the glomerulus. So it's like a renal artery comes, then it starts to become thinner and thinner and thinner, whilst it's going round and round and tying itself into knots, because it knows if it can get as thin as it can get, when blood is flowing through those thin tubes, it will be at high pressure. And if it's at high pressure, then we are going to have things fall out. Things fall out, okay? Particles fall out of these vessels from the blood. They are in the blood, but they are going to fall out. And these particles that are falling out, what are the particles? glucose. Tell me, what else comes out of here? Students, tell me the things that are going to come out of your glomerulus and fall into the Bowman's capsule. We have glucose, then we have water. Thank you. Then what else? Excess salts. You can just say the salts. Okay, just say salts because the ones that are in excess will stay in the urine. So just say water. Jen here, just say salts. And what else? Urea. These things are falling out into the Bowman's capsule. Bowman's capsule will say thank you. Then they start moving like this. Once you get to the PCT, then what happens? Glucose will be reabsorbed. Whoom! At the very first bend, the PCT, glucose is reabsorbed. Right? Students? Hello? Once you get to the proximal, then convoluted, it's a convoluted tube, it's a bend. Then glucose goes. Then what goes at the loop of Henle? Tell me, what is going to be reabsorbed at the loop of Henle? It had fallen out of the blood, but it will be reabsorbed back into the blood. And what is it? Class, you're too quiet. Just a quick revision. What is reabsorbed here? Yes. At the loop of Henle, we have salts. What else? Water. So salts and water are reabsorbed here. Okay? Then at the DCT, anything? Anything at the DCT? No. What happens at the collecting duct? More water goes. That's it. Now, if you are going to have glucose go all the way and it stays and it doesn't even get reabsorbed and we find it in the urine, it doesn't get reabsorbed back into this thing, into the blood here. It doesn't go back into the, it should go back into the blood. If you have glucose staying in the urine, because the urine is what will keep moving. This is urine, eh? Urine is what will keep moving, what will keep moving. So when we say we have urine and we find glucose is not gone, it has not been reabsorbed back into the blood, okay, it's not gone back into the blood, the glucose, then we have a problem. It means that your glucose was in excess. If glucose was in excess, not all of it can be reabsorbed. So some will be found in the urine. And if you have excess glucose, then we know that your pancreas didn't do its job. You have diabetes of some sort. Are we together? Either the that would be type 2 diabetes. Okay, that's it. So here, this is what you'll be talking about. This is what this is just talking about. It's talking about the same thing. And it talks about the things that remain in the blood. Okay? Urea remains in urine. It is not reabsorbed. Water is absorbed at the loop of Henle and at the collecting duct. Glucose at the PCT. Salts at the loop of Henle. Urea will not be reabsorbed. It will be pushed out at the glomerulus into the Bowman's capsule, and urea will stay. Urea will stay in the urine forever and ever and be thrown out because it is toxic. We don't even want it. All right, good. I'm happy. Yeah, it's good. It's something. At least there's something. Now we are going into the very last part. Okay, we're going into the very last part of excretion. And I'm just going to talk about, let me write with my thick pen here. Can you write the role? Write with me, I'll write fast. The role of the liver. The role of the liver in excretion. The role of the liver in excretion. That's what I want you to write. Let's quickly move through this. Now, this is the same thing that I'll have tomorrow. It will be just a subheading like this. The only difference is that it won't be my handwriting because I'm going to do it with my PC tonight, and then you get the notes. Let's go. Now, the question is, why do we even talk about the liver in excretion? Why is the liver in excretion? Huh? Kidneys are the ones that remove waste. Why are we even talking about the liver? Right. Why are we even talking about the liver in excretion? Why is it there? Let's write here. Can you write for me? Here is the answer. The liver makes, okay, it is the one that makes one of the major products that the kidneys remove or filter out of the blood. The liver, we know when blood gets to the kidneys, it will push those things through ultrafiltration at the glomerulus. It will push water, salts, glucose, urea out of the blood. But nobody told you where the urea comes from. Okay. This waste product is urea. Urea is produced by the liver. Urea is produced by the liver. Students? Really? Yes. How? You need to know how. Okay. Urea comes from proteins. Okay. Comes from proteins. Really? Yes. Okay. When we eat things like one, I know you haven't done digestion, I'll remind you, okay? But there's meat, there's eggs, there is beans, there is milk, okay? All these are serious proteins. These foodstuffs. When you know digestion, you must know the balanced diet. That's the first thing. And when you say you know balanced diet, you know the 7 essential nutrients, and you know which foods give you those things. So there's milk, then there's fish. So it's very easy to look at your food and know if you're eating a balanced diet. Remember, it's the right foods in the right amounts. Okay? Perfect country. So when you eat things like this, okay? Proteins. These are proteins. These are protein sources. Okay? You're going to get proteins from these things. Did I put fish? Yes. Right. Now, let's see what happens. Proteins are then digested into amino acids. The proteins are broken down and they make amino acids. Amino acids, of course, these are the building blocks of proteins. So this is like reverse. Huh? We're breaking down into the parts. Now, okay, and so put in brackets when you say proteins are broken back into amino acids, say building blocks of proteins. Now, do we have a problem? Yes. Why do we have a problem? The body cannot store excess amino acids. Okay? The body can only store excess glucose and fat. Yeah, the body can store glucose and the body can store fat. Yeah, it doesn't have a problem. But don't ever give the body excess amino acids. It will have to deal with the excess amino acids. So, right to say, if you eat more protein, you eat too much meat or milk or whatever. If you eat more protein than your body needs, then all the extra amino acids must be dealt with. Don't let me down now. You know that the moment you eat proteins, your body will digest them back to amino acids. But these cannot be in excess. So let's say you ate too much milk or whatever. Now you have amino acids in your body. Well, the body will use the amino acids it needs. Now it has excess. What can it do? Can it store them? No. So what does we know that when we are doing coordination and response, when your body releases a hormone, when the response is done, that hormone must be removed. Now, a hormone is a protein. Who removes it? The liver. Whatever hormone is released into your blood should be removed when the response is done. And it's the liver that does that. So obviously, when we have this second situation, where we have amino acids in our blood that have come from us eating, the liver must come again and do the same thing it did to hormones when it was done. When the hormones were done, the liver should deal with this. So what does the liver do? The liver removes excess amino acids from circulation. The liver will remove the excess amino acids from circulation. Write this, please. I want my notes. Right. Are we together? This is what leads to deamination. So this is what leads to you needing to know something called deamination. Are we together? Right. Deamination is like it's removing the problem. The liver will remove the problem. What is deamination? Let's talk about it. This is the removal of the amino group from an acid. Right. Amino group, please put in brackets to say it is NH2. Okay, amine. Nitrogen is there. I know you're not yet in A level where you study amines, but just know amino group is a nitrogen, ammonia, amino, NH something. So okay, are we together? This is the amino. This amino group is going to be removed from your amino acid. Okay? Students, are we together? Of course, your amino acid, if you're asked to show, your amino acid should have like this. Let me just show you. It'll be a carbon. Let's just say a carbon at the center here. Then this side is your carboxylic acid. Then this side is the amino group. Then we don't care how many carbons it has. So this is like the central carbon. You cannot forget this. This is so cheap and easy. This is the amino group, and this is the acid. So from this amino acid, we want to remove this. And go and let the other thing go. Because amino acids cannot be stored. So we need to change them. So now let's do the steps. Steps of deamination. What is deamination? Removal of amino. To deaminate, you are removing the amino group from an amino acid that was made from just digesting a protein. So the moment you digest a protein, it's obvious you are coming back here. Because this is what makes proteins. It's not abracadabra here. So you have come back to this amino acid. Now you have a problem. So now you got to get rid of the amino group. That's what the liver will do for you. The liver will produce urea by doing deamination. Come on now. You ain't going to let me down. Step 1. What it does is say excess amino acid. Okay? The excess. Because, of course, your body needs some to make some proteins. All those hormones, whatever you need, enzymes, all these things. But then there are some that will be in excess. And those excess cannot stay in you. Things don't just stay in the blood. When the body doesn't need them, they have to get out. So excess amino acid enters the liver. The liver will deal with this excess amino. Keyword, class, keyword, excess. Keyword, excess. Excess amino acid enters the liver. Then the liver removes the amino group from the amino acid. The liver will remove the amino group from the amino acid. Then step 3, ammonia is formed. Ammonia is formed. Step 4. What remains is this carbon skeleton, the acid, whatever. So I would say the remaining carbon skeleton is left behind. So basically, we are not done. Thank you, liver. You have taken a nuisance from the blood, excess amino acids. Because of course there will always be excess. We don't know how many, when to stop eating protein. We'll be just eating, we don't know. Now, this amino acid problem has been solved. We no longer have amino acid in the blood excess, okay? All of it has been converted to ammonia. So should the liver rest now? No, it cannot rest. This is impossible. Let me tell you the new problem we now have. This ammonia is a problem. It can damage all your cells. It's a toxic thing. So ammonia is toxic. So it's a dangerous waste. So this ammonia should be even further converted now. So the liver should now do the second. But here it is done with deamination. So deamination is done. Don't put another step here. It's no longer deamination. Yeah? No. That's another problem. So now let's now skip 2 lines and say we have created ammonia. Now, ammonia is dangerous waste. Okay? Because it is very toxic. So the liver, please don't let us down. Don't let that ammonia out, liver. No. It's very toxic. Okay? Even small amounts can damage cells. Even small amounts of ammonia can damage cells. So liver, act quickly, please. I want you to write this way. Let me take a red pen. Get rid of this ammonia, Mr. Liver. You have not solved the problem fully. So the liver quickly converts ammonia into urea. Okay? Urea is not food. The liver converts ammonia into urea, but still this urea will need to be thrown out of the body. But it's not as toxic as ammonia. Okay? So this is because urea is much less toxic than ammonia. Do not forget this. So this is what happens. Excess amino acid goes into the liver. There is deamination. Ammonia is produced. Then the ammonia is converted to urea. Okay, it converts ammonia to urea. Urea then enters the bloodstream. Okay, it is less toxic, so we can let it get back into the bloodstream. But forget about letting ammonia back into the bloodstream. That can't happen. It gets into the blood, enters into the bloodstream, and the kidneys now will start excretion, and the kidneys remove urea from the blood. Okay. Urea then leaves the body in urine. We saw what urine has. Urine has urea and excess salts and excess water. Okay, it shouldn't have glucose because all the glucose should be quickly reabsorbed at the PCT. Are we together? Right. Now, let me just tell you something. The liver, something about the liver, okay? When the amino group is removed by the liver from amino acid in deamination. The remaining carbon skeleton, we call it, you can say the remaining carbon containing part, carbon skeleton, is not wasted by the liver. It is not wasted by the liver. Okay? The remaining carbon skeleton, it means carbon containing. When we say carbon skeleton, it's just something that is carbon. Okay? Is used in respiration and some converted to glycogen and stored. And some will be converted to glycogen and stored. Okay? Are we together? Students, are we together? Right. Give me a second. There's one last part I want to give you, then we stop this lesson. Give me 1 sec. Okay? Let me just attend to something. All right. Thank you. Let me just give you what I wanted to give you. I didn't want to stop the lesson without giving you this because it's the end of excretion, so I don't want to do it again. In the next lesson, we'll be talking about something else. Just want to give you, remember? Let me give you some tables, okay? Should I give you a table? Let me think. Okay, let me tell you this before I give you. Urea should be excreted. Class, to be excreted is to be removed from the blood. So this is something that was in the blood. We're not talking about food that you eat and it doesn't even get into your bloodstream. We're talking about things that were in the blood. That is why excretion is not egestion. Excretion is talking about things that are being removed from the blood. These things go into urine. Don't try to force your own thinking into this. We only talked about urine and we ended there. So urine is things that are coming from your blood. Feces is things that are coming from your stomach. Don't try to force nothing. Thank you. Urea should be excreted. Why? Even though urea is less toxic than ammonia. Have you ever heard of people who've had a kidney failure? Kidney failure is a big problem because now it means that urea is no longer being removed from the blood. And let me tell you what happens. Let me write it, okay, instead of even though it is less toxic than ammonia, I don't want to explain. It is still a waste product. Okay? I need to save time, so I should not explain. I'm doing more of giving you information that you can write down because I know you will use it. I know you will use it. If I explain, then you will hear me today, then what tomorrow? Urea is just as bad. Ammonia, in small amounts, it will destroy your cells. Okay? Ammonia is more toxic, which means when you get even a small amount of ammonia, it will damage your cells. When your cells are being damaged, that's dying. Okay? Because what you are made of is cells. So you can't say, it's just my cells dying. It's you. You're made up of cells. So when your cells are dying, it's you dying. That's like cancer, which kills cells, right? So when we say we have toxic things, they're killing your cells, which means they're killing the person. Urea also does the same thing, but it only happens when it builds up. When the urea builds up into amounts, then it's damaging your cells. So I'm going to say, if too much urea accumulates, then the cells become damaged. And the body does not function well. The body will not function. Okay? If urea accumulates, it will damage cells. So it must be removed from the bloodstream. And what is that? Excretion. Who does that? The nephron. It is a part of what? Of the kidneys. Well done. Must be removed from the bloodstreams. Must be removed from the bloodstream. By who? By the nephron. Which would be in the kidney. So we can also say by the kidneys. Okay. Anyway, I'll give you some questions that you must answer, and we'll see how it goes. Now, next chapter is going to be reproduction. I'm going to teach you the difference between sexual and asexual reproduction. Then I tell you, we'll talk about reproduction in plants, the stages, the pollination, the fertilization, the seed formation, blah, 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 things like that. Then talk about reproduction in animals. No, not in animals, but in human beings, okay? Which is similar to what we already know, gametes, the fertilization part, then the pregnancy, those cycles, we talk about it, and the hormones, the sexual hormones, okay? We'll talk about the menstrual cycle as well. And lastly, I think the last thing which you have to talk about is STIs, sexually transmitted infection. It's part of reproduction, so you have to know it. Very important. So get yourselves ready. We're going to have fun. Okay? So we'll talk about in plants and we'll talk about in animals. We'll talk about the parts of a plant, and I'll show you. You must never ever forget them. We cannot afford to lose marks. And I think the last chapter which I want to talk about is drugs. Then we'll do a lot of questions because you'll be knowing a lot of chapters for now. Then we can go back to the older chapters and we'll talk about them. Okay, we have a lot of work to do in these 3 months. So come on now, we are not going to stop. Excretion, we are done. Is everybody happy? Is there anyone with a question? Okay, you're happy. Good. So we move on to reproduction in um, reproduction as our next chapter. Let me just take the attendance so that I can put a bit of pressure on the students who dismiss themselves. Good. Right. Next is business studies. Those who don't do business studies, you are dismissed.